how much I needed to learn about all this, even though I'm not a decision maker. I'm looking forward to hearing him speak. He's part of the Department of Veterinary and Biomedical Sciences at the University of Minnesota College of Veterinary Medicine. I told him Madam Chair is a grad of UMN, and that is about all he knows about this meeting other than it's informational. So thank you for squeezing us into your busy schedule, Professor Larson. So why don't you take it away?
Thank you so much for that introduction. Can you all see this screen okay?
Mm-hmm.
Yes.
Okay, great. Let me kind of rearrange stuff here on my screen so I can see everyone. Very excited to present before you today. I was asked to cover some basics about chronic wasting disease and a little bit about the current research that our group at the Center for Prion Research and Outreach is doing.
You can hear back there. Is there a way to get sound back there?
Can we turn? Is there a volume button? Is there a volume button here?
Wanted to have Heidi come in and do it.
I'll have Heidi come.
A little quiet over there.
I'll have Heidi come.
We can have fun with this.
So that Professor Larson can stay on his timetable. That would be great.
All right.
So should I hold off until the volumes?
Yeah, just give us a second and see if we can resolve this volume issue.
Okay. I can try to keep talking so you can tell if it really is. Actually, up here in Bemidji, our team is, many of you are aware or heard of the Beltrami.
That was
Sorry?
We found the volume. We're testing it.
you hear it?
Okay,
All right.
a little better?
Yes. Thank you.
Okay.
Does it have independent volumes on these sites? Because
They
this
couldn't hear down there.
Yeah.
Is it,
You know.
should I say the alphabet or?
Sing the rouser.
Okay.
You all don't want to hear me sing. So up here in Beltrami County, we have a diagnostic test that allows us to look for prions, CWD causing prions in soil and water. And we're using that test to look at the Carcass dump site here in Beltrami County. And I'll talk more about that at the end of my slides. But I wanted to go through just a basic introduction of CWD, what a prion is, how it can become infectious. It's just going to help us understand this disease. And I've been down to the Preston area, Winona area, several times over the last 3 years to work alongside the DNR in their surveillance effort and down in that part of the state. And so, my hope is going through these slides that we'll have a better understanding of the disease itself. So this is the outline. What are these prions? Most of us have heard of these prions, but they are mysterious. And so I want to try to do a better job of explaining that to everyone so we have a better understanding of what prions are, how they can be of concern with the ecology of Minnesota, especially with our waterways. And then I will talk a little bit about the advancements that our team at the Prion Research and Outreach Center has made in diagnostics. So CWD is a progressive, fatal, 100% fatal neurodegenerative disease of cervids. Most of us know this. It's not caused by a virus or a bacteria. It's actually caused by a misfolded prion protein. And I have slides that will help describe that. The disease was first identified in Colorado in the 1960s. It's thought that mule deer were kept in the same pen that with sheep that had scrapies. Scrapie is a prion disease of sheep and goats. And so over time, it's thought that the mule deer that were in that pen, their prions started to misfold after being in contact with those scrapie infected sheep. And then it got out of that facility and started to spread in Colorado. And you can see this map. I don't know how the slides are coming through on your end, but the dark counties here in Colorado are kind of where the epicenter, the start of CWD was in the 1960s. And all these other gray counties are where it is found today. It's currently found in like 26 states, up here in Canada, real bad. All the yellow dots are areas are farms that have been depopulated. The red dots are farms that have CWD and have not been depopulated. So still some out in Wisconsin and whatnot. You can see the Beltrami farm up here depopulated in April. Okay. So it's really important to understand that chronic wasting disease, it belongs to this whole family of prion diseases. So these are collectively called TSEs, transmissible spongiform encephalopathies. They actually, these prion diseases cause the brain to essentially look like a sponge with over time. You get these large, they call them vacuoles, but these holes essentially in the brain where these prions are, and that causes nerve death. And I'll describe a little bit more about that. But this is related to scrapie, as I said, but also BSE or mad cow disease in cattle, and then Creutzfeldt-Jakob disease or CJD in humans. So we know a lot about these other diseases that helps us understand about CWD, which is part of that family, part of that spectrum of prion diseases. So what is a prion? And when I teach about this at the University of Minnesota, I'll ask the students, how many have heard of a prion? Most everyone raises their hand, and I say, how many feel comfortable like knowing, how do you actually know what a prion is and how it becomes infectious? And everyone lowers their hand, right? Because going through school, we did not get this education. The field of prion biology is really new. So all mammals have prion proteins. And I'm showing you a ribbon diagram here, a structure, a three-dimensional structure of what, this is how scientists can help view proteins. And this, when I'm on the right there, that's just a normal prion. All mammals have prions, humans have prions. They play an important role in regulating metals in the body. It's the same for deer. So they help these prion proteins, normal functioning prion proteins, regulate copper, other metals. They have important functional roles in nerve cells. Okay, so we all have prion proteins, and the way to visualize this, think of a slinky. Think of a slinky, a three-dimensional object slinky. If I had a slinky here with me, I'd be moving it back and forth. Think of that like a normal prion protein. A misfolded prion protein, the one that causes CWD or CJD or BSE or scrapie, looks like this on the right. It gets all misfolded and it starts to stack up. It creates these sticky, gummy plaques, almost like Alzheimer's disease, like amyloid. So when it misfolds, it creates all these sheets we call them beta sheets. So it changes its three-dimensional structure. So think of a slinky. If I take that normal slinky and I just ball it all up and twist it up and everything, it's no longer functional, right? Like it won't act like a slinky acts. It would just roll down the stairs. It wouldn't glide down the stairs. So the misfolded prion that causes disease, that's infectious, is like that misfolded slinky. It doesn't work anymore. It's changed its shape. And they grow, they stick together and they start to form plaques and tangles and almost grow like crystals inside of cells. They just stack together like this, okay? So if a CWD prion, the misfolded form, comes into contact and touches a normal prion protein, let's say a deer is somehow ingested the misfolded prion protein, if it gets into its GI tract, it could come into contact with other normal proteins. When they touch, it causes it to misfold. So just the act of one of the misfolded prion coming into contact with the normal one, it interacts and it causes the normal one to misfold. It's like a domino effect or a daisy chain. Okay, so this is how, like at the cell, like when a deer is exposed to CWD, what's happening at the cellular level is a domino effect. It starts to propagate, it touches, and it causes the others to misfold, and it just replicates over time. So I think of this like it's the chaos engine of CWD, because all it takes is a few of the misfolded prions to come into the animal. They start touching normal prions, and then those go and touch normal prions, and they convert, and it goes boom, 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 boom. That process from when the animal is exposed to when you see a phenotype like on the right here, that can take 2 or 3 years. So you can have a deer that has this happening, the daisy chain happening in its body, but it could take 2 or 3 years before you see it at that late stage. An animal that's had the infection maybe for 3-6 months, a year, will look perfectly healthy. Okay, it's just that these prions are slowly propagating, misfolding within the animal. It takes that long, 2 years, for them to colonize the brain and start to kill neurons. So what's happening is, typically we think that the most common route for CWD to enter deer is like by ingestion. Imagine, like, if you have, I mean, deer are social animals. They're licking, grooming each other, bucks fighting. And then also I'll talk about how prions can actually remain infectious in the environment, like in the soil, for up to 16 years. And so it's thought that the most common route is ingestion or inhalation. So once the prions get in, they slowly start to grow, like propagate. The domino effect is happening. It's spreading through the nerves. There's big nerves that join from the stomach all the way up to the brain. So this is like a highway for these prions right up to the brain. It takes about 2 years to get up there. On the right, I'm showing you a slice of American cheese. This is a slice of just normal healthy brain. On the bottom, a slice of Swiss cheese. This is what a CWD brain looks like. All those white holes are big spot, like these are areas where nerves are dead. They're gone. Just gaping holes. This is how the spongiform appearance for these prion diseases, their brain looks like a sponge, because the misfolded prions are causing nerves to die with time. I always get asked this: do humans get CWD? The answer is that there's no confirmed cases that humans can get CWD from eating contaminated venison. However, there is evidence that other species can acquire prion disease from being exposed to CWD causing prions. These are typically challenge experiments where they actually take the prions and inject them straight, like into the animal's brain or whatnot. Like if they do this, cattle can get it, but it's like a direct injection. There's no evidence that cattle can get it just by consuming, right? Or being in farms where CWD has been documented in the past. There's some concern that pigs can acquire CWD. I think there's experiments from the USDA showing like 30% of their pig challenges, the pigs actually got a prion disease. There's some species of monkeys. There's going to be a paper coming out showing that macaques eating positive venison acquired a prion disease. So it's concerning. And even though there's no direct evidence of it causing disease in humans, the scientists, including myself, are concerned about exposure, because the reason is, if you think about that slinky, that misfolded slinky, it has different shapes. That slinky, that prion, can misfold in different shapes. And it's possible that there's some shape of a CWD prion that, if it misfolds a certain way, when it comes in contact to a human prion, it might be able to cause it to misfold. It's just that we don't, as the disease spreads and more people, it's estimated 15 to 20,000 positive CWD positive venison carcasses are being consumed every year. That is going to grow, like, with time as CWD spreads. And so this is why diagnostics is so important, that we can try to prevent the prions from entering our food supply, because we don't want to find that one prion combination that can fit with the human prion. So we know the BSE outbreak, prion disease, that happened in the UK in the 80s and early 90s. 177 people died from that. They were eating contaminated meat. And those prions, the cow prions that are misfolded, they are a good match for humans, and they do cause prion disease, variant Creutzfeldt-Jakob disease. I was actually down at Winona State, and I gave a seminar there before the pandemic, and so there was someone in the crowd that came up to me afterwards, and he was in tears. He said that he had a family member who died as part of this outbreak of mad cow disease. So we know that the cow prions can cause humans to convert. Scrapie, on the other hand, that's been known since the 1700s. No evidence at all. There's a lot of mutton consumed around the world. There's no evidence that Scrapie has caused prion disease in humans. We think that CWD is more towards Scrapie and that it's not, causing disease in humans, but the reality is we don't really know. As the disease spreads, it gets into new populations of white-tailed deer, other cervids. What is that prion diversity like? Strain diversity, we don't have a good idea yet. So these prions, these misfolded prions, they can be spread in bodily fluids, direct or indirect contact. So the prions themselves are found in saliva. They're shed in saliva, blood, urine, feces. Our diagnostic test now, we can use blood and feces from live animals, and we can detect the CWD prions there. We have a live animal test. Those prions can remain infectious in the soil for years. Up to 16 years has been shown for Scrapie. It's been shown in research in Colorado for CWD up to like 4 years, 5 years, but no one's really tested beyond 5 years, right? The science hasn't been done yet to see what's the maximum end of how long CWD prions can remain infectious in the environment. Scrapie prions have been found in dust. And so we're going to be doing some research looking at, can we detect the CWD prions in dust? And that might be a way for other deer to inhale, if you will, those prions if they're bound to dust particles. And as we know, yes, deer are very social animals. What's concerning is that those misfolded proteins, they have been shown to experimentally to actually be able to be brought in through the roots of plants, and the plants will deposit the prions in the leaves. And this is a picture in the lower right here where there was an experiment done where they grew grass essentially in CWD prion contaminated soil. Those CWD prions got deposited in leaves. They took a leaf clipping and fed it to a hamster, a rodent model that they do their research on, and that rodent died of a prion disease. So they showed experimentally that prions could be uptaken into plants. We're aware of a paper that's going to be published in the next 6 months to a year that shows in a lab setting that prions can actually be uptaken into alfalfa and corn. And so when I talk to legislators about this, I talk about the looming issues this means for agriculture. One country is already, Norway, has already banned the importation of hay or straw from any CWD positive region. All hay or straw imported into Norway require a letter from a vet saying that these were not raised where CWD is. So I view this as a sleeping giant to the egg economy in the United States. So environmental contamination. It's really, really important. All the discussions at the legislature about carcass disposal and landfills, it's really important because if you think of an animal that's died of CWD, that there's a lot of misfolded prions there. We are testing carcasses. We believe there's been 9 to 12 carcasses positive for CWD dumped at this Beltrami site that we're working. We can detect them, and we can detect them in the soil. And as I said, the deer can output these into the environment through their bodily fluids, but also scavengers can spread those around the environment. Okay, so here in Beltrami at the dump site, we're seeing a spine, a skull, a scapula dragged across 12 acres. Well, we're seeing that positive, we're getting positive, hits across a wide geographic area. It's because of the scavengers. And yeah, they can remain infectious in the soil for years. It's been shown experimentally. What about water? This is something that I was asked to talk about. Yes, the misfolded prion, that misshapen one, since it's resistant, like when it takes that different shape, it does not degrade easily. It can withstand, you cannot cook it. The only way that you can destroy it is by using like 50% bleach solution, and that only works for stainless steel. It can just remain intact for a long time. So because of that, you can detect CWD prions in water. We know this from research out of Colorado. There's a team that detected prions in snowmelt runoff from a positive herd of mule deer on the landscape. They just sampled the water coming off that during the spring, and quite simply were able to filter that and then detect the prions in the water. They also detected prions at the intake valve of a water treatment plant. Okay, and so there's concern there is where are these prions? We don't really have a good understanding. Some of this depends on the frequency of CWD in the wild. Right now in the southeastern part of the state where y'all are, it's like 1 to 2%. That's still very low. This is why managing the spread of disease is so important, because you don't want to have herds with a 50% positivity rate outputting these prions in the environment. They're just going to be going all over the place, if you will, when thinking about water dispersal. We know that the more organic water, material in water, the longer the prions remain infectious. The prions, they can bind to metals and they bind to organic material in soils in different ways. Some of the binding can actually make them more infectious. Some can make them less infectious. The bottom line is we don't have a good understanding of what is happening in Minnesota. We're just starting to research now. So there's zero data on how these prions are circulating in Minnesota's waterways. On the map, I'm just showing you, and then the most recent ones will be up here in Beltrami. There are point sources. So it's unfortunately every positive farm that every Serpent farm that goes positive, depending on when that farm was found to be positive, the longer that herd is on the landscape, the more prions are outputting into the environment, the more that accumulate on that spot. So you can think of it as almost like a point source, right? And so the question is, what's going on downstream? And so we're going to be doing research. We can go and sample downstream of some of these locations to see, can we detect them or not? We don't know. Also in the southeastern part of the state, as I said, where some of these herds are, the infection rate is still low, 1 to 2%. But if we were to go down in the area where y'all are, if we were to do some sampling from some waterways downstream of, I mean, the whole area is heavily populated by deer, but would we be able to find some prions? Don't know. We're going to start that. So we have received funding now from the state. It's going to be a 3-year project where we can go around and target areas and get water samples and then test them to see if we can detect prions. Just because we detect prions doesn't necessarily mean they're infectious or will cause disease. There's other experiments we can do to determine that, but surveillance is the first step. So how can we stop the spread of this? Hunting, hunting, hunting. It's so important that that we keep hunting pressure up. That's been shown in other areas of the United States, that is, if hunting pressure goes down where areas where CWD is, CWD can go up. So that plus the work that the DNR is doing, the culling that the USDA does alongside the DNR, all that is very, very important, because you have to keep those, the incidence of CWD at a low level in order to manage the spread more effectively. We have to develop new tools. So some of you are aware, my group, we were funded about 2 or 3 years ago, about $2 million to develop new diagnostic tests that can be used in a variety of situations, both in the environment, but also live and harvested deer. And we're doing that, and we've had great success. I think that pre-movement testing is really important. We can do that now. That's an option. It just takes action at the state level with the Board of Mental Health to be able to take the tools that we have access to and expand them. MinPro is the center that we've put together, developed in 2019. Dr. Tiffany Wolf is co-director of MinPro along myself. Mark Schwabenlander is associate director. We have this new diagnostic test. It's called RT-Quick. And if you haven't heard about this, you'll be hearing more about it in the near future. This is an assay. This RT-Quick is an assay that's so sensitive. If you were to take 1 tablespoon of those misfolded prions, it'd be prions, and mix it in 400 Olympic-sized swimming pools, this test is that sensitive. We can pick them up using this test. This test leverages the way that the domino effect that I talked about. We can take a tube of normal prions that we grow up in the lab, non-infectious, normal, they're grown with bacteria. We can take a little sample, let's say it's of, I don't know, water that we've filtered and we want to test. We put it in there, You just incubate and shake, and in about 12 hours, the reaction, that domino effect, if the misfolded prions are there, it'll spread within the tube, and then we have a fluorescent dye that binds there and gives a signal. That's essentially how the test works, and it's very reliable, very sensitive, low false positive rate. So this is the primary workhorse in our Prion lab. We have 6 machines, process at least 144 samples every 48 hours. We have protocols now for venison, muscle. We were thinking about this as a food safety test, working with the Minnesota Department of Ag, but also protocols for plants, water, and soil. Okay, a little bit towards the end here on our research in Beltrami County. So, and this is, again, where I'm at right now. We're gonna be out in the field tomorrow collecting samples. But in April, doe tested positive. There's a trace back to Winona County. This is like over 200-mile dispersal. If there was a pre-movement test, if we had that live animal test, which is functional now because of RT Quick in our lab, if that was scaled up and embraced by the state, the Board of Animal Health, we could prevent this situation from happening. We could actually test these animals before they're moved and say, no, these are positive. We could test essentially we could test all the farms, I believe, within the state. That helps stabilize the turbid farming community, also helps prevent CWD from spreading because of the situation. There was a dump site that was discovered through the investigation that the state performed. We were asked by the DNR to get samples. You can see the samples there on the bottom. The herd was depopulated in May. There was 13 total positive deer out of like 55. Most of those are fawns. Fawns can get CWD. Fawns can be born with CWD. There's a misnomer out there that where people think that fawns can't get it. They do. It takes 2 years for it to kill an animal. So a fawn could be born, look perfectly healthy, but it has CWD. This is a view of the fence that was erected. So in June, the DNR erected this tall fence. The dump site was on forested land, so they had to go through and log around all the way around 12 acres, erect this fence. We were able to identify multiple bone and insect samples. So we collected fly larvae that were on some of the carcass remains, actually inside the bones themselves. We tested those fly larvae, and the bone marrow was positive, so were the fly larvae. They were positive too. So with those prions, we can track just by the animal or the insects, if you will, that are feeding on the carcasses themselves. We also took soil cores under the bones when we picked them up, and after we identified that some of those bones were positive, we tested the soil. Soil is positive. So the prions are like leaching out of the bones into the soil underneath. And so now we're back here to take more samples and see what how far these prions are moving in the environment. And I think it's about 12 to 15 acres of what this fence has been erected. So we work with a great diversity of folks all across the state, tribal. We have national research partners. There's like over 70 MinPro research associates. So we're working with a large group, both at the state level, local, state, and national level. Because of the RT Quick technology, we're recommending live animal testing, ecological testing. I even think that we could sample tank or feces of herds themselves and do a herd level surveillance just by sampling the water, the stock tank. Our value system, MinPro's value system, we really embrace that land grant institution. And so part of my job is to work with the state, to work with stakeholders on things like CWD and try to find new solutions to this critical problem, because it's going to impact our state economically quite a bit in the coming years. So thank you for your time. I'm happy to answer any questions that you might have.
Questions?
Yeah.
Go ahead.
So as far as the test of live animals, you're saying that we need to sort of gear that up? And is that on the back of the legislature? Is that who needs to make that happen?
Yeah. Yes. So it's the Board of Animal Health.
Yeah.
Right. They have language in their CWD regulations that say any test deemed acceptable, I forget what the language is, by the Board of Animal Health can be used. It doesn't say, so there's a difference here. USDA validated test, that's something that comes from the national regulatory, right, from the USDA. But the state can take something like this RT Quick, and it can be used as a live animal test. But it needs to come from the Board of Animal Health, the legislatures. The MinPro lab we are the R&D lab. We can come up, and we didn't develop RT Quick. RT Quick's been around since 2007. More and more labs are getting access to it. We're the only one in the state that has it right now. But we can, the veterinary diagnostic lab at the University of Minnesota, they process thousands and thousands of samples. We can get this up and running in the BDL. And so this is a viable option, I think. How long will it take? Maybe 2 years. But if we start now, we can get there.
But you just triggered, if it's a viable option, so I don't understand. If you shoot a deer out in the woods, what's the timeline and how do you do this if this equipment is only at your lab? What does the hunter do?
So we're not a diagnostic lab for the state. So that's the thing. So all hunted deer right now, like what the DNR does for the CWD sampling, they take all those samples, tissue samples, they ship them out to Colorado or Wisconsin to labs that have the personnel and they're not using RT Quick. They're using things called ELISA and IHC. These are the tests that are the traditional tests. The results for that can take anywhere from 4 days up to 4 weeks. It all depends on all the samples, thousands of samples coming into the main diagnostic lab. RT Quick, the technology, we have it, available in our lab. We're a small research lab. We've shown how it works. We've tested it out on many samples. It generates results within 24 to 48 hours, depending on the sample type. It's a viable option because we've already done the groundwork, if you will, to show that it is functional and can be used and can be scaled up. What would need to happen if the state wanted to expand it, more funding would need to come in, more people would need to be hired, more machines purchased to be able to handle the thousands of samples, for example, on the hunting side of things. For live animal testing, it's a different ballgame. There's in the cervid farming industry in the state, there's about 250, maybe 300 cervid farms. It could be used as a live animal test, and I've presented that to the Board of Animal Health. And again, the correct way forward would be for the veterinary diagnostic lab that employs lots of technicians who could run the test. They would need to get this up and running. Our lab would teach them essentially how to do it, and then we could expand from there.
Else? All right. No other questions. I appreciate your time and information, and take care of Beltrami County. They're friends of ours, so.
We'll try. We'll do our best. Thanks again. Thanks for the opportunity. Happy to answer any more questions if you have via email.
All right. Thank you.
Thank you.
Thank you very much. Our next speaker tonight is Todd Frober. He's the acting Big Game Program Coordinator, the Fish and Wildlife Division of the Minnesota DNR, the Whitewater Wildlife Management Area. And I know that you folks know Todd. He's spoken before. He also has some slides that I think Heidi is so on the ball always. So, so please welcome Todd.
You introduced me, I guess, I'll give a slightly different presentation, obviously, than Peter. He focused more on the, I guess, diagnostics of CWD. I'm just going to talk about what we're planning to do for.
Sorry, just quick interruption. If you could just face this direction because of that will probably work better. Thank you. Sorry to interrupt.
Sorry, don't worry about my back. So.
And if you can take the mask off while you're speaking, if you like to.
So I'll just give I guess an update about what we're planning for surveillance and management for, I guess, deer regulations for 2021 for fall. So next. Is that happening in here or outside?
Just ask Heidi to. Okay. There you go.
Thanks, Heidi.
So I guess the current status of CWD, so since 2002, we've had a little over 90,000 deer sampled. And most of the positive wild whitetail deer have come from Fillmore County. As you can see, 81 deer and 20 from Winona County. In Houston County, there's been a small localized area, basically on one property where all 5 of them positives have come from. And there's another cluster of positives in Dakota County, so it's south of the cities. That was actually on that last slide, that deer that was standing, that was in the clinical phase. So it's a cluster around there. Olmsted County, one was from the old Elk Run farm. There was a deer that was found positive outside of that positive farm, and then there has been a detection there since. And then there was actually one found within the Rochester city limits last year, which presents problems in itself due to restrictions on hunting and such. And then also the Crow Wing County, which was surveillance surrounding a positive farm. There was another whitetail deer or wild deer found outside the fence. And there has not been a detection since then. So hopefully, fingers crossed, this will be the last year of that being a management area. And then there's been 12 positive Serpent farms in the state. So 3 elk, 8 whitetail deer, mixed deer, and 1 redtail deer farm. The latest one being what Peter mentioned was Beltrami County. Next. So samples for fall 2020, just to review, with COVID, it kind of minimized the staff stations. So it was voluntary sampling and self-service sampling stations set up around mostly in the southeast and south metro, but our management zones, and there was 27 positives found, all of those being in southeast and south metro. Next. I guess the detections of CWD positive deer and wild deer, there was 27 cases last year, 24 of them being in southeastern Minnesota, so 16 of those positives from Fillmore, or from fall hunting, 4 positives from opportunistic, so vehicle killed or found dead, so reported, and then 4 positives from agency culling. And then there's three in South Metro. The CWD management zone, 1 from the fall regular hunt, 1 from the fall into winter late hunt, special hunt, and then 1 from agency culling. And then, as I mentioned, that north central management zone, Crow Wing County up around Brainerd, there has not been any positives found since the original detection. So, next. And Peter mentioned this as well, the apparent prevalence of CWD, and this is in our deer permit area 603, 647, 48, which reverted back as the Fillmore County area. So apparent prevalence is still less than 1%, so it's pretty rare. It's important to keep in mind. Next. And the overview for this fall. Those dark yellow areas are the CWD management zones. Those are where carcasses cannot be taken out, so the carcass movement restrictions. Those light yellow areas are control zones. Carcasses cannot be taken out of those areas, but can move into the management zones. And then those surveillance areas, the gray ones, they're not carcass movement restrictions, but in all 3 of those zones, there's mandatory testing for the opening A and B seasons, and then outside of those zones, there's voluntary testing. So either via them self-service sampling stations in the management and control zone. Otherwise, for the surveillance zone, there's voluntary testing by a wildlife area office.
So essentially, it's a quarantine. It's a quarantine in those dark yellow areas.
Yeah, carcasses cannot leave those DPAs.
And is that statewide internally, or can we stop those carcasses from moving south into Iowa? Do we have that authority?
We do. They cannot leave those DPAs. They cannot leave that management zone, so they can't cross into Iowa.
But
And
isn't
we
most
have
of that Houston, isn't it most of it Houston County?
Other part, 649 is roughly mostly Houston County.
But I thought you said Houston County had like 5 and Winona County had 20. So why are we telling Houston County they can't move their deer?
It's if there's a wild positive detection within the DPA, so that DPA is encompassing our management zone.
How many deer farms did you say there are in the state?
I believe there's about 170 with whitetail deer. There's, I think, somewhere between 250 and 300 Serpent farms.
Okay, so, I'm sorry,
How many
I
in
should
Houston and
understand that.
Winona?
What's the difference between a whitetail and a Serpent farm?
Well, Serpent is the overarching, I guess, deer species are,
Oh, elk or something?
elk, moose, deer, red deer, whitetail deer, mule deer, so.
Okay, and in an earlier slide, you showed a picture of a deer you said was positive and it was outside the fence.
Yeah, so, I mean, until recently, we've been granted co-management of whitetail, specifically whitetail deer farms, not any of the rest of the Serpent species, with the Board of Animal Health. But before that, it was just, we had regulatory authority over wild whitetail deer, so.
So when you say the fence, I assume you're talking about a Serpent farm fence, or what fence are you
Correct,
talking?
yeah.
Okay, and so was there positives in that Serpent farm?
Sorry, which,
I'm just trying to figure out for sure, are you 100% confident that the farm deer are the problem that are spreading it to the wild deer? Or if there's a wild deer on the outside of the fence, could he be giving it to the Serpent farm? I mean, which way is, I'm just trying to understand what you're talking about when you say there's a deer outside the fence that's positive.
yeah, so in that, I mean, that farm specifically, I think in Crow Wing County, he had pretty high prevalence. I mean, most parsimonious explanation is that that wild whitetail deer got it from the farm. There's probably other cases where farm deer are getting it from wild whitetail deer. There's no, it's not like a virus that has strains. It's not traceable to a certain region. So it's not like avian influenza or something where you can trace it to a specific region. So it's unknown, to be honest. So.
Okay, so now you said in the dark yellow, and that's primarily Houston County, and is that one up to the upper, to the left, is that Winona County? It's hard to tell which
Yeah,
map
sorry,
is.
the map is kind of small, so.
Is that part of Winona County? That one
Yeah,
circle?
so 646 is.
That's Winona County.
Yeah,
And all the others down in the,
mainly, yeah,
okay, and
Those
all
don't
the other,
follow county lines.
no, they don't follow county
not
lines.
quite. Yeah,
No,
well,
but the
major
whole
highways.
other county. And I thought, I'm sorry, I thought there was a big problem in Fillmore County. So is
Yeah.
Fillmore County
Yeah,
represented?
that's 647, 648.
Okay, well, you're assuming I can read those numbers,
sorry.
your chart is. So that's a good portion of Fillmore and Houston County. So you said you can't remove, so until the deer is tested or
Yeah.
or what?
Until it's tested, not detected by that ELISA test that Peter mentioned.
Okay, and how, from a consumer meat safety, how long, can these animals be kept? I mean, do they have to be kept in cold storage then, or?
Well, I guess. if
Depends
you
on the
have that
weather?
option, that'd be
And
ideal,
how about the
but,
trophy hunters? Can they take the head or the antlers? Or is
they
it
have to leave
everything?
Carcass in the head within the zone, so you can cape out the deer, which is just if you have a skull plate and clean the brain tissue off, which is how, a mount is done.
Uh-huh.
So it's just the hide and then the antlers.
So they can take those?
Yeah, and then we have dumpsters
Okay,
scattered around the areas for them to dump the rest in, or if they're, like what I do, if I shoot a deer, I quarter my deer and I deposit the carcass and the head within one of them dumpsters, and then I freeze my meat until I get my test back before I do anything else with it, so.
and you said, I think the other gentleman said that part of the management is hunt, hunt, hunt. So are people allowed to take more deer in this area than normal?
Yeah, there's pretty liberalized regulations within these management zones, so.
Okay, but I, as a landowner, can do more, or as a recreational hunter can do more, or both?
Yeah, all of the above. Yep.
Okay, all right.
So is each area that has a number, it can't be moved from inside of that boundary? Or for in southeast Minnesota, is it the red line that is?
Those red lines,
It's
yes.
the red lines, okay,
600
yeah.
series is the management zone, 300 and that 255 is control, and then everything outside of that is surveillance or.
So you can
not
take it from
involved.
Winona, from Fillmore to Winona, but you can't take it out
Correct.
to Mower probably.
Yep. All right, next, I think. So I think there was one before that, but I can, yeah, so it's just, there's 87 stations total. And I guess within those zones, as I mentioned, it's mandatory sampling for opening A and B season, the opening weekend, because that's obviously when we get the majority of the deer shot. And some of them other stations, there's about 30% of the stations that will have self-service sampling stations. So if someone doesn't feel comfortable in the COVID landscape of providing a sample to a staff, they have the ability to just drop their head off in there or provide those samples. Next. I guess the one thing that did change for southeastern Minnesota was it used to be unlimited antlerless deer. It's a reduction of the bag limit, so it's 5 deer within the management zones. It does not apply to the early antlerless seasons or the late season hunts. And this is actually the first year that those CWD late hunts are included in the regs book. So we do know that when those will be, that's a weekend, 3-day weekend before Christmas and the 3-day weekend after Christmas. So that would actually be New Year's Eve, New Year's Day weekend. So, and then the deer feeding ban will be expanded to include for the positive farm detection in Beltrami County. Next. And this program was, I guess, legislated and mandated in 2019. So we began this on a wider scale. Those are all the locations of where the dumpsters are. These are DNR provided dumpsters for hunters to provide their carcasses. And last year it generated about 200 tons of deer waste. So it's one very tangible and kind of visceral way for hunters to kind of try to control the spread of CWD and not move those carcasses around. Provides that option to get rid of the carcass. And
what does the DNR do with those?
Those are hauled to line landfill, so it's a approved method of disposal. So they're clay line landfill. So
here, okay, so if you shot one in Winona County, where's our closest clay line landfill?
they actually go to La Crosse. So
we're transporting them across state lines.
yeah, we asked Wisconsin and they were fine with it. So they either go to La Crosse or Olmsted County landfill.
When you say carcass, is that all that, when you shoot the deer and you got it out, the guts and everything, is that all going, or what's happening with that?
No, so the carcass movement restrictions includes the spinal column and the head. So any brain tissue or any part of the spinal column. So
I thought this last gentleman just explained that it could be contained on any part of the animal and that it could spread to the environment.
Yep.
But you can gut them and you just would do it. You dress it out like you normally would and leave that lay.
Yep. Unfortunately, yeah, there's no perfect solution for it. It's just kind of,
But, so you think that there's a higher concentration of it in the brain and spine, is what you're saying?
yeah, in the central nervous system is where this prions accumulate. So that are lymphatic tissue. So like I say, it's not a perfect solution, but you can only control so much. So
Seems like some more research needs to be done there. That's a little disturbing. Lay all the internal organs just there and leach into the soils and predators. We got coyotes and turkeys and everything else coming around. Farm dogs.
Your dog.
Yeah,
Yeah,
farm dogs and it just.
it's a give and take. If you put too many restrictions on hunters, they're gonna drop out of the ranks and either not participate or, not participate in hunting or not participate in the
It's kind of
program.
like the
So,
little boy putting his thumb in the dike in Denmark, trying to stop the water. Okay.
Next. And then, I guess what's required of the hunter, so register your deer. Just like always, we have mandatory registration in Minnesota. And then comply with those mandatory testings on opening weekends. You can give it, you can put your deer in one of them self-service sampling stations, see if you're outside of those weekends for voluntary testing. And then don't take those intact carcasses outside of the CWD zones. Next. And as we discussed, and we kind of hit on this, of those, what is composed of within the carcass restrictions, that's the head and the spinal column. Any of those positive deers or positive material go to alkaline digester at the University of Minnesota. If we have them, we can't catch all of them, but a lot of them have gone into those dumpsters, which, as I mentioned, go to those landfills. So that proper disposal is key. This is an area where hunters can do their part and try to contain the spread of CWD. Next. And as Peter mentioned, they're up in Beltrami now. This is just another picture of that fence that was constructed. This was a positive deer farm, and he had knowingly or unknowingly dumped, I guess probably not knowingly dumped positive carcasses, but knowingly dumped carcasses on public land. So those costs were, I think actually the final number was $196,000. Next. And that's it. So
Are other, you said hotspots in Colorado and Canada, are they following similar
yeah,
protocols?
there's 26 states and 3 provinces, and that's an unfortunate result of, I guess, our legislative boundaries. Is each state somewhat has different authorities within their game agencies, or how they handle it, or what's granted to them, I guess, by their legislature, or kind of what the abilities they have to do, or ultimately funding. So
Are they doing something similar, or is it
there
very
are some
different?
states, yes, that are, and there's some states that are not. So
And success rates, are you comparing?
it's tough to determine success with CWD. There's not a lot of success. Success is kind of, I don't know, I guess you use air quotes when you say success, because success is pretty much keeping prevalence low. There's one thing that seems to be pretty clear: the states that employ active management can keep their prevalence low. There still is radial spread, but their prevalences are low. Wisconsin is pretty much given up, and it's hard to blame them, because they've had it for quite a bit longer, and they were kind of at the forefront. They didn't there's more unknown than there is now, and they have faced some pretty hard political decisions versus biological decisions, and they essentially gave up the fight on CWD, and I mean, their prevalence is exponentially grown, and in some areas it's as high as 40 to 50% in adult males, and 30 to 40% in adult females. So
We have deer that travel from Wisconsin or from Iowa, yes.
oh yeah, we have collars on deer that over in
They don't
Fillmore
travel
County.
far though, do they?
Some of them have. There was one doe. I mean, these are pretty, I guess, anomalies, if you will. But there was one doe that traveled 76 miles from, I believe she was collared down by Harmony, and went all the way up by Cannon Falls. There was a couple other ones that went pretty far into Iowa, about 40 miles. Those, I think, both were juvenile males this last year. So there are some, and there's been observations of deer from, it's actually a old DNR employee that did his master thesis on deer in the Whitewater River Valley that documented deer traveling to Wisconsin. So
But
there's
usually
no
they're,
question that they're
they do,
traveling back.
but as a general rule, they're a herd and they stay in a,
Typically, yeah, I
somewhat
mean,
geographic area.
and adult males have smaller home ranges than juvenile males. I know
Yeah.
they're getting kicked off those home ranges.
Sure.
And then there's, at, you know, and different
Teenagers.
different times of the
Just
season,
teenagers.
there's dispersals. So those does move to fawning grounds and they herd up in the winter. So
Mhm. Okay.
But with success being defined as keeping the prevalence low and knowing that around us we have animals that are infected, we're just trying to manage this at this point.
we're trying to manage, contain the spread, contain, and keep prevalence low until, I guess, science catches up to where we're at. So,
Trying to keep it low, and then meanwhile, we're testing to make sure to see what's happening with the water.
yeah, that's, they're doing more of those diagnostic tests. DNR surveillance is more for, I guess, distribution and prevalence on the landscape. So, and that's, of course, obviously where we're doing antemortem tests, so, or postmortem, sorry.
So you said that hunter can get up to a permit to take 5 deer in these areas. Is that what I heard you say?
Yeah, it's five deer limit, and that does not include so there's an early antler season where you can take an additional 5, and then there's late season where it's actually unlimited either sex. So there really isn't a limit, and that, I mean, ultimately there's not there's not a real restriction for somebody if they're really looking to take deer. So
Isn't the deer populations still expanding even with?
yeah, I mean, I don't, there's not a shortage of deer. I think we have pretty high densities in most of southeastern Minnesota. So, and some areas have, you know, a lot higher localized densities wherever the habitat is. It was more of a, I mean, I don't want to say just a perception issue, but I mean,
Are the numbers increasing because there's less hunting? Is that, I mean, I live in the Whitewater area, and when I was younger, I recall literally cars lined up and down the country, rural road. And now, you know, we'll see, I probably see a fraction of, you know, maybe 2 or 3, 4% of what I saw when I was a kid.
yeah, I mean, I think
It's not because all these farmers say no, it's just, there just doesn't seem to be the interest anymore.
yeah, there's I would say a multitude of reasons. There's less hunters, land use changes that's made it more preferable for deer habitat, forestry changes. And I would say ultimately from the time I was a kid till now, deer hunting culture has changed drastically. Most people will only target mature bucks and not shoot any does at all. So they're doing essentially nothing to manage the herd. I would say that we're living a lot easier, living a lot higher on the hog nowadays than people were in probably the 70s, 80s, and 90s. People just don't shoot deer like they used to. I think the average hunter now shoots 1.2 deer. So your
It's just, it's a tradition that's not being passed from generation to generation, like it once
Yeah.
was.
Then people just don't eat deer. I think people, I mean, before my generation, my dad's
Well, us
generation,
farmers have made
shot a
beef
lot of
awfully
deer.
tasty.
Yeah, well, that's right. But I think the saying used to go was beef was for the market and venison was for the table. So I think a lot of those farmers used to shoot a lot of deer. So,
But you said in your case, you quarter your deer, freeze it until you get your test back. You won't eat a positive deer?
I personally won't. I guess I'm not willing to assume those risks. I know people have different risk tolerances, but I have young kids, and if the incubation period in humans is, I guess, 10 to 20 years, I mean, I'd be pretty devastated if I fed my kid infected venison and then they got sick. So everybody's risk tolerance is different. I'm not going to tell someone they can't. We don't tell someone they can't. The CDC recommends that they shouldn't. I'm not going to personally. I don't need to. So that's just kind of my take, I guess.
Right.
And then, like Peter said, there seems to be in-lab tests that show that there's potential, and they're just knowing the TSE family that there's potential for species barrier restrictions, but also some species jump. So the risk is not zero, so. It's all like probably pretty low, but I guess the more times we take the chance, the more times there's risk, so.
So maybe you should take a piece of the steak and have it tested as well as the spine and the
Yeah,
head.
so that we're testing is your lymph
Yeah,
nodes, so we have the same, like when you're sick, they swell up and are sore, so that's kind of the first place of prions accumulate is in your central nervous system and your lymphatic, because it's fighting off disease or fighting off infection. So it's the most.
It's a guessing game. to a
Yeah, I
certain-
mean,
to a certain degree.
that's been tested of where they can find, and that ELISA test is not as sensitive as that RT Quick is what Peter's using to test some of the soil and water and other parts of the deer. So that's kind of why theirs is way more sensitive than that ELISA test. So.
Okay. All right. So are we getting an RT Quick in southeast Minnesota?
Probably at some point. The science is developing pretty quick, but I wouldn't bank on it for this season or next season or the
Well,
season after.
what are the priorities of the DNR?
Well, I think that funding is going way over the DNR's head. So
All right. Okay. Thank you.
Thank
Thank you very much.
The Bluff Country chapter of the Minnesota Deer Counters invited their state director, Perry Fitch, and I apologize, Perry, I didn't get a chance to meet you beforehand, but I wanted to give him a chance to talk about his organization. He was one of the groups that was recommended to join this work session. And in addition, the Farm Bureau was invited and accepted, and Glenn Groth sends his apologies and regrets, but the farm called today, and so he canceled today, but with a promise to participate in future if asked.
All right.
Thank you, Perry.
We understand it's harvest season.
Well, I'm Perry Fitch. I live out by Pickwick. Some of you I know. So anyhow, Minnesota Deer Hunters Association, I'm the state director for the Bluff Country chapter. We have roughly 400, 450 members, primarily from Winona County, Houston County. I'm not sure what you guys want me to talk about CWD. I'm with Todd. I'm not eating one. I'll have mine tested before I eat them, and I harvest a lot of deer. I do have a farm, so but we do get them all tested. I've had one positive out of my deer that we've shot. So actually, we were probably the first one in Winona County. So that happened. It was in the extended season in the wintertime. It's not that I like to deer hunt, but it was about 15, 20 below that day, and I was hunting. So, I love to deer hunt. The CWD thing bothers me. Not as much me as I'd like my grandkids to hunt. And I don't think kids will hunt if the population goes to the 20, 30, 40% of the herd. And I'm sure that's going to happen over in Wisconsin. I'm guessing it is. But here, it's not yet. I would recommend to the county, I'm not sure what county land is available for hunting that you allow hunting on, whether if it's in a season place where you can only use bows, let them use bows. Let them use guns where they can. But wherever you can allow the hunting on county land would be, much appreciated by the DNR and the Minnesota Deer Hunters Association as well.
When you say hunting, are you talking about general population, general public hunting, or are you talking about the DNR sharpshooters?
No, I'm talking general public. Let the people hunt for you.
Okay.
The DNR costs money to hunt. I'm just saying. And they, not that they don't have a budget, they do, but it's our money going to the DNR to pay for them to hunt. So let the public hunt as much as possible.
Does your organization, have they taken a stand and officially said they support all the restrictions or whatever that would come
We
with?
support CWD restrictions. Yes, we do.
Okay, as an organization.
As an organization statewide.
Okay,
Yes.
all right. Because when this came up last time, we had some individuals very much opposed to, some of the,
Oh, and I'm not saying it's 100%. I'm
right,
not saying.
but as an organization.
But as an organization, we do support the CWD we support the DNR and all their attempts at doing what they can to keep the positives down.
Okay, all right. And the question was asked earlier in the observation of the lack of hunters. And of course, you indicated, yeah, CWD might, if it gets high, would deter you from, do you think that is impacting our deer hunting currently?
I believe it has impacted. I would say 1 or 2% so far, maybe more. You could take it at, maybe Todd has the deer hunters numbers. I know when it first hit, it went down quite a bit, but I think it did come up. Todd, can you elaborate on that?
There's some parts of Minnesota that seem to have it, but I don't know if it's attributed strictly to CWD, but there's also in Wisconsin, there's decreased numbers of different CWD areas that there's been a problem.
So are most of our deer hunters in the Winona area, are they local people or do we get a lot of influx of people coming from other parts of the state to deer hunt?
Ballpark guess on me would we got about 80% locals.
Local?
10% relatives and 10% from other areas. That's just my guess, but as my observation is getting older and watching things.
Sure.
You know.
Okay. All right.
And it does come back to, as a kid that started hunting in the 70s, we hunted for food.
Right.
We don't hunt for food no more. Food's too available.
Yep.
So it's a biggie. So.
Okay. And is the, you encourage the hunting on county-owned land. More and more landowners are restricting the amount of hunting.
Some are.
Some
No
are.
question about it. Some people lease their land for trophy hunting, and stuff like that. That happens. It's money to be made. But yeah, just if the county in general, we are a very good county for deer, would kind of promote their hunting like, hey, whatever. And I don't even specify a county land, but I know there's areas that the county owns that hunting's
Sure.
not allowed.
Yeah.
And archery is pretty successful rate, and you'll get some people complain, I'm sure.
Okay.
So,
Okay. I would like to enroll the deer in a safe avoidance of car class.
car avoidance, that's
Yeah,
another one.
yeah, we need
Which,
to teach them.
some hunting helps out with that.
Teach
some.
them to stay off the roads at night when I can't see
Well, at
them.
least teach them to cross by the sides.
Yeah, deer crossing. They do that in Canada.
So you did state that you support all the DNR's measures to try to control the chronic wasting disease. So 2 years ago, we were approached at the county board to allow DNR sharpshooters on county property. And then we were approached again last year, kind of at the last minute, and I don't know if we'll be approached this year. The first year we allowed it. The second year, the public outcry was quite a bit greater. And so I'm wondering what your position is on that. I did hear you say very plainly, allow the public to hunt.
Right.
What are your thoughts on the DNR's sharpshooting?
I call it like a necessary evil. I mean, that's what it is. We have certain areas in the DNR is well aware of the areas where there's probably a high probability of positive deer, and as far as I know, they concentrate on them areas.
So your advice?
I'd rather have the public harvest the deer.
If the DNR had a time where they're sharpshooting, if there was alternative times, you would say open it to the public as well?
Yes.
So they're just not out there at the same time?
Right. No, you wouldn't want them there at the same time. No, no. But just the public, just in the regular seasons, just let them hunt that land. I mean, wherever it's even feasible. So.
All right. Any other questions for Mr. Finch?
Nope. Encourage your grandkids and kids to hunt. Like, take them hunting.
All right.
Right.
Thank
Okay.
you.
Thank you.
Thank you, everyone, for attending. So we'll have the slides made available to everyone as well as the public. And if there are any questions, you can get back to our speakers and so on. Otherwise, you have a break before the meeting. You might need a little break.
Okay.
So otherwise, thank you very much. And
All right.
I certainly learned a lot. Folks might be better informed than I, but I really appreciate the opportunity to sit in and do it as well. Thank you.
All right.